Welcome to my talk, and thanks for giving me the opportunity to present my work here in the PhD Graduate Talk. And yeah, today I want to tell you something about my PhD work about the effects of mucilage and extracellular polymeric substances on soil gas diffusion. And yeah, as to why should we look at soil gas diffusion processes? So we know the population is growing and climate change will increase the water scarcity, which will have a great impact on the food production, and therefore optimized resource use in agriculture is crucial to address these challenges. And there you go. And yeah, an optimal plant growth requires both, so water and an adequate supply of oxygen, and therefore a profound understanding of the soil aeration and respiration processes is needed. And the main movement of oxygen is via diffusion, so diffusion from the atmosphere into the soil towards the root. But before the oxygen can reach the root, it needs to pass the rhizosphere, so it's a small layer of soil around the root, which physical properties are actively modified by mucilage. So mucilage, it's a polymeric hydrogel-like substance that is secreted by the root tip during growing and depositing around the root, creating the so-called rhizosphere. But it's not only mucilage is secreted into the pore space, also bacteria produces similar polymeric substances called EPS, so extracellular polymeric substances that are also deposited into the pore space. And if we take a closer look at both, both are so-called viscoelastic hydrogels, that can hold large amounts of water. So depending on the type, it's up to 600 times its own dry weight. And both form these liquid structures between soil particles during drying. And the breakup of these filaments, they depend on the viscosity, the surface tension, the radius of the filament, and the density. And if we now take a look at current models to describe the diffusion through the rhizosphere, we see that they treat the mucilage as a uniform layer surrounding the root. So this would mean that if the oxygen now diffuses towards the root, it needs to diffuse through the liquid phase. And the diffusion through the liquid phase is up to 10,000 times slower than the diffusion through the gas phase, which then would result in a reduction of the diffusivity by a factor more than 1,000. And if we take a look at soil gas diffusion models in general, we can see here a general formula to describe the diffusion, and we can see that the air-filled porosity is the primary variable controlling the soil gas diffusion. Yet we have seen that mucilage in EPS forms this more like filamentous structures in soil, which might favor oxygen diffusion compared to a continuous bulk mucilage layer. And also, if we imagine this as these thin filament structures, they might also not affect the air-filled porosity. So this leads to the main question of my research. So what is now the influence of mucilage and EPS on the soil gas diffusion? Therefore, I addressed 4 main questions. So how do dried mucilage patterns affect the soil gas diffusion? Does the effect of mucilage on gas diffusion depend on the particle size? Does the hysteresis we know from the soil water characteristic curve during a drying rewetting cycle also affects the soil gas diffusion? And how does mucilage influence this effect? And are the similarities between mucilage and EPS also represented in their effect on soil gas diffusion? So to answer the first question, I developed a conceptual model here. So on the one hand, we can see it as a single root with the rhizosphere surrounding or whole root system. And the hypothesis here is that at low mucilage contents, we have just formation of this thin filamentous structure where gas can easily diffuse by, and this will only have a small effect on the gas diffusion. While with increasing mucilage content, larger structures emerge. So we have the cylindrical structures between the soil particles, which can now clog the pores, increasing the diffusion pathways, and by this reducing the gas diffusion. While we expect that high mucilage contents, these structures also are interconnected, span over multiple pores, and will have a significant impact on the soil gas diffusion. In addition, I used the numerical simulations to describe the formation on the one hand of water and on the other hand of the mucilage structures during drying here between two soil particles. And here we could see for the liquid case, so we have this behavior of a water bridge that breaks at low water contents, while for high mucilage content, we have more like a elastic network. And here we could see the formation of this cylindrical structures that are actually also hollow inside. To visualize these mucilage structures, I used environmental scanning electron microscopy. Here you can see images from a sandy soil mixed with shear seed mucilage, and it was dried for 48 hours. So water content water content was below 1%. And we have an increasing mucilage content from left to right. So 1 milligram of mucilage, of dry mucilage per gram of soil on the left, and then increasing up to 5 milligram per gram of soil. And here we can see for the low mucilage contents, this formation of these filamentous structures between the particles. And with increasing content, we could see these cylindrical structures then between the soil particles. And at the highest content, you can here see the structure spanning over multiple cores, looking like a foil through the pore space, which then will block the diffusion pathways. And to test this hypothesis, I performed a diffusion chamber experiment to determine the gas diffusion coefficient. Therefore, I used the setup you can see here. So it's basically the body of a desiccator, and on top an acrylic plate with an opening for the soil sample. And I used two contactless oxygen sensors to measure the amount of oxygen inside the desiccator, and I placed it on a magnetic stirrer to make sure that we have a homogeneous distribution of the air inside the chamber, so to have an accurate measurement. And I used oxygen then as a tracer gas. Therefore, I initially flushed the chamber with nitrogen to remove all the oxygen. Then the soil sample was placed on the opening, and the oxygen then starts to diffuse through the soil sample into the desiccator, and then I measured the increase in oxygen inside the chamber. And at the same time, the nitrogen, of course, diffused out of the soil sample. Used for this shear seed mucilage as a model for plant mucilage. I used artificial soil, so it was grinded quartz sand here to a particle size of 500 to 630 micrometers. I packed them in PVC tubes with the given properties, and the samples here were air dried for 48 hours at room temperature. For the results, here we can see that with the increasing mucilage content, the diffusion was decreased. We could then assume here, if we have a mucilage density of roughly 1 gram per cubic centimeter, that the volume occupied by mucilage in the sample was less than 1.04% of the volume of the bulk soil. So we could see here a reduction of a factor between, I would say, 2 and 3. And this was without affecting the air fluid porosity. So here we can say that the effect of the dry mucilage on the air fluid porosity was then negligible, and the diffusion was reduced then only by this dry mucilage structures. So in the next step, I wanted to know if the effect depends on the particle size, and also if the hysteresis of the soil water characteristic is also represented in the soil gas diffusion, and how mucilage will will influence this effect. So therefore, I developed the following conceptual model. So we have an increasing particle size from left to right and an increasing mucilage content from bottom to top, all at a given water content. And here the hypothesis is that we have a decrease in gas diffusivity, of course, with an increasing mucilage content. For the coarse-sized particles, higher mucilage content is required for the structures to persist because we have rather large pores, and the breakup of the filaments depends on the radius then of the filaments. And for the finer-sized particles, we expect reduced gas diffusivity due to the higher number of potential pore throats. So we have a higher number of pores, but and at the same time a smaller pore radius. And here we also then expect for the fine-sized soil hysteresis effect, same as for the water retention characteristic. So during drying, water can still be held in narrow parts of the pore space, here indicated in the red cycle. You can also use the laser pointer here. While we have during the rewetting phase, if we have a rewetting due to capillary forces, water might not be able to reach these parts, and they will still be open during rewetting for the gas to diffuse through, while they are blocked during the drying. And with increasing mucilage content, we expect that the interconnected mucilage structures will then also be able to attract water again during rewetting here in parts where it might not reach without the mucilage, and diffusivity is then again decreased, and the hysteresis effect will diminish. So short summary then of the hypothesis. So decrease in gas diffusivity with increasing mucilage content, and for the coarse-sized particles, higher mucilage content will be required for the structures to persist. And in finer-sized particles, the higher number of potential pore throats will then lead to a reduced gas diffusivity and hysteresis that will diminish with an increasing mucilage content. Here again, performed the same measurements as before, now with different particle sizes. And also here we used in the shear seed mucilage as a model for plant mucilage, and slightly different in the sample size here compared to the measurements before. And again, they were tried for 48 hours at room temperature. And if we first take a look at the results for the dried state, so again for 48 hours of drying with a water content of less than 1%, we have again the relative diffusion coefficient on the y-axis, mucilage content on the x-axis. And if we start with the silt and clay-sized particles, here we could see that the addition of mucilage had no effect on the gas diffusivity, even at high mucilage contents, which is most likely to, we have here a high number of pores. So mucilage is not able to deposit in every pore, and this will at sometimes overweight the potential pore throats where mucilage can accumulate. So we have still a lot of pores open for the gas to diffuse through, and mucilage will have, or does have here, no effect. And if we now increase the particle size for the fine sand-sized soil, we can then see that with the addition of mucilage, the diffusivity is reduced again, and we can see the highest reduction in the medium-sized soil, a medium sand-sized soil, where we have a reduction of a factor of 6. And for the larger particle sizes here, the coarse sand, the diffusivity increased again, but is also then decreased with an increasing mucilage content. So also to summarize, for the fine particle size, we have a higher number of pores where mucilage cannot deposit in every pore. And we have the highest reduction here for the medium-sized sandy soil with a factor of 6. If we now take a look at the results during a drying rewetting cycle, here we start with the control soil, so without any mucilage. We have on the x-axis, again, the relative diffusion coefficient, on the y-axis, the volumetric water content. And overall, first we can see a reduction of the diffusivity with an increasing water content. But if we look closer at the fine-sized particles, we could see that we here have a hysteresis effect. So we have a higher diffusivity during rewetting compared to a lower diffusivity during drying. And I want to draw your attention now to this value. So for the 10% volumetric water content during the drying phase here for the fine-sized sandy soil particles, I will now add the mucilage. So we will go step by step, increase the mucilage content, and we will start with 0.5 milligram of mucilage per gram of soil. And here we can see that the hysteresis effect starts to diminish. And what's more, surprisingly, we have an increase in diffusivity during the drying, despite while adding mucilage. So intuitively, you would think we add this gel-like substance, and I was more expecting that diffusivity during rewetting was decreased, but here was the case that the diffusivity increased again during the drying phase. And if I add more mucilage, so we have now 1 milligram of mucilage per gram of soil, and here we could see that it really increased during the rewetting phase, during the drying phase, now matching the value during rewetting, and the hysteresis effect diminished. And with the further addition of mucilage, with 2.5 milligram per gram of soil, the diffusion is reduced again. And you can see at the highest content that even in the dry phase, we have here for the fine-sized sandy particles, the same effect as the addition, or almost same effect as the addition of water here at the volumetric water content of 10%. And here we can also see again that the addition of mucilage for the dry phase had no effect in the silt and clay-sized particles. So what happened? So a possible explanation I have is that mucilage absorbs the water and starts to swell, and this is affecting the soil structure. So during swelling, it pushes the particles away and open up the pores again for the mucilage to diffuse through. So if we look at the red cycles, the pore here is closed without mucilage, water is held in there, then the gel starts to swell, pushes the particles away, increasing the, let's say, pore radius. Now at the low water contents, the mucilage is not able to span across these pores to block them, and they are now open again for the gas to diffuse through. And only at the high mucilage contents, now the bridges can again persist in the pore space and block the diffusion pathways again. Here to say in the clay-sized samples, like the clay-sized particles here, I suppressed the swelling effect because during the sample preparation, I noticed that you could really see and measure an increase in the height of the soil samples. So the swelling resulted in a higher soil column compared to the others. But to have comparable results, I want to keep them all at the same level, at the same height. And therefore, I compressed the clay-sized particles slightly, and with this, suppressed here the swelling effect. Or it was not as then in the results as distinct as in the fine sand-sized particles. Here to summarize here the findings. So for the fine-sized particles, we had a higher gas diffusivity during rewetting compared to a lower gas diffusivity during drying. The effect diminished with an increasing mucilage content and increased gas diffusivity during drying at low mucilage contents, which indicates that the soil structure then is affected by the gel. So in the next step, I used the X-ray computed tomography to analyze the connectivity of the gas and the liquid phase on the soil samples. I used here again shear seed mucilage as a model for the plant mucilage. Here again, the artificial soil, so the quad sand here with 200 to 500 micrometer particle size. And here I packed them in sample holders with a height and a diameter of 1 centimeter. So really small samples. But I had to do it, or why I did it was that if you take a look here at the picture, how the imaging works. So we have the X-ray source, we have the object, and we have the detector plane. And the closer you can get the object to the X-ray source, the larger will be the image on the detector plane and the higher the resolution. So we had small, to cover the whole sample, it needs to be quite small and close to the X-ray source. And with this, I could reach a resolution of 11 micrometers. And here you can see a example, so a slice from the reconstructed image. And to analyze the images, I used the image software Viso, which was quite nice because I could write a Python script and that fully automated the image processing and the analysis, which was quite helpful regarding the large amount of samples I had to analyze. And here for the pre-processing and the analysis, we have again the reconstructed image. So it's a grayscale image. And I segmented the image, which means to each gray value here is referred to one of the three phases in the soil. So here the dark gray values are referred to the air phase here in light blue. The light gray values then are represented here in dark blue as the liquid phase. And here the rather bright samples are the soil particles are here represented in brown in the segmented image. And what you can get, for example, is a 3D image of the connected gas phase throughout the whole sample. And here again, you have a image, a pixel size in the image of 11 times 11 squared micrometers. Now to quantify the connectivity, I used two well-established metrics. We have on the one hand the Euler-Poincaré characteristic. What it does, it counts the number of disconnected components or clusters. In this case here we have two. Then it's minus the genus. Genus means redundant connections. So we have 1, 2, and 3 redundant connections, so minus 3. And then it adds again the completely enclosed cavities. So here in soil samples, they are usually neglected because a completely enclosed cavity would mean that you have a soil particle surrounded by air, so that which means the particle is levitating in the pore space, which makes no sense. And what you get is a number, and the more negative this number, the better is the connection. The other metric is the gamma indicator, so it's a connection probability. So it's the probability of two voxels belonging to the same cluster. So it reaches from 0 to 1, where values close to 0 is a bad connectivity and or a low probability to find the connectivity, and values close to 1 is a high probability to find connectivity. And as for the results, we have here for the Euler-Poincaré characteristic on the y-axis, and again the volumetric water content on the x-axis. We have the control soil and 1 milligram per gram mucilage content and 2.5 milligram per gram mucilage content. And in the top row, we have the gas phase, and the bottom row, the liquid phase. And also here we could see Yeah, first of all, at low water contents, negative number for the gas phase, which makes sense. We have a good connectivity of the gas phase, and high values for the liquid phase, which makes also sense. So it's close to zero water content. So we have a bad connectivity of the liquid phase. And here we could also see for the gas phase, let's say, indications of the hysteresis effect that is diminishing with increasing mucilage content. Yet here we have to say we have here, of course, limits in resolution. So these fine mucilage filament structures, they could not be detected with the CT imaging at this resolution. And also during the image processing, the images were cropped to to have the same sample size. So we cut off a bit from the top layer and a bit from the bottom layer, which also might be the areas with the most water content, or which might have altered the soil moisture content. And this is why it also results in these large error bars. And here the results for the gamma indicator. Also, if we start here with the control soil, increasing mucilage content like before, we could here see for the rather dry state, we have values close to 1, so a high probability to find the connected gas phase, and a low probability to find the connected liquid phase, which makes sense. And also here again, slight indications then with the increasing water content of the hysteresis in the gas phase that seems to diminish with the addition of mucilage. So to conclude or to summarize, for the gas phase connectivity, we have the indication of a higher connectivity during rewetting, lower connectivity during drying, resulting also in the hysteresis effect, which also seems to diminish with increasing mucilage content. And here we have to keep in mind that we have limits in the resolution. So the thin filaments, they were not detectable, which might also have a reduction in the connectivity of the gas phase. So this leads us now to the last my last research questions. So are the similarities between mucilage and EPS also represented in their effect on the soil gas diffusion? And the hypothesis was yes. So I expect due to the similar formation of this liquid structures between the particles and the similarity in the basic traits, they will have the same effect. So again, the diffusion chamber experiment to determine the gas diffusion coefficient like before. And here I used extracellular polymeric substances from a Rhinobacillus polymyxa and the Bacillus licheniformis. So both are known for their existence in soil, or especially the rhizosphere, and both are known for their high EPS production. And for this, I mixed them to a content of 1 milligram per gram of soil and packed them again to the same sample size as before, also tried at room temperature. And as for the results, we start again with the control soil like before. And now with here with the hysteresis again at this point. And if we add then 1 milligram of EPS from Bacillus polymyxa per gram of soil, we could here see the same effect, an increase for the drying cycle and diminishing hysteresis. And same could be observed for the EPS produced by the Bacillus licheniformis. So short summary, similar effects to those of mucilage were observed. So to come to an end of my talk, I want to summarize the findings from my thesis. So we have seen that mucilage forms these liquid structures in the pore space depending on its content. It reduces the gas diffusivity without affecting the effluent porosity, here up to a factor of 6. The reduction in the gas diffusivity depended on the particle size, so could observe no difference in the dry soil, for example, in clay and silty-sized particles. Depends on the mucilage content and, of course, on the water content. And we have a diminishing hysteresis in the gas diffusion and in the gas phase conductivity. With the addition of mucilage, and for EPS, similar effects were observed. So a bit as a conclusion and an outlook, so accurate prediction of soil gas diffusion is crucial for optimizing resource utilization, irrigation strategies, and ultimately the crop performance. So we have seen for the gas diffusion model, we cannot treat mucilage as a uniform layer, so it forms these reticular structures where we would expect a reduction by a factor more than thousands if we treat it as a uniform layer. I could show that the highest reduction by this mucilage structures was by a factor of 6, and reduced airfield porosity as a primary variable for the diffusion models is not reliable here if we add mucilage, so we should more think of an additional tortuosity factor we need to implement in the models. As for the mucilage and DPS interactions, you have seen that, this mucilage and DPS, they maintain the stable conditions here regarding soil moisture fluctuations. So we have seen diminishing hysteresis in the gas diffusion with the addition of mucilage and DPS. And also we can think of stable conditions regarding if the bacteria in soil degrades the mucilage, but also produces this EPS. It compensates then the degradation of mucilage by the production of EPS. And so we have also along the route stable conditions over a longer time. Of course, there were a lot of limitations here in my studies and open questions. So we are still at the beginning of this research. So I used seed mucilage from the chia seeds. Also here it would be the next step to also do this for the for plant-derived mucilage. Also I used here artificial salts. The study work done under lab conditions. Also the resolution in the X-ray CT imaging can be improved. And the next step and the big open question would here the effects of mucilage and EPS on soil structure, which might then also explain the observed increase in the gas diffusivity during the drying phase with the addition of low mucilage contents. And to somehow give a vision where I want to end with this. So if we know our conditions, that at some time we then can use an appropriate amount of water for the fields to have then an optimal use efficiency of the water at the best crop performance. And with this, I want to thank you for your attention, and I'm open for some questions.